Hello everyone, welcome to this webinar on biomedical engineering simulations with COMSOL Multiphysics. My name is Jennifer Segee, and I'm a technical marketing engineer working out of the Burlington, Massachusetts offices of COMSOL. I'm presenting today together with Dr. Mark Yauman. Mark, the founder of Continuum Blue, has a degree in engineering and a PhD in computational modeling and applied mathematics. His postgraduate work focused on the design and optimization of cardiovascular implants for Medtronic, Inc., using finite element methods and genetic algorithms. With over 15 years of experience in the field of multiphysics modeling, he has worked in a wide variety of industrial sectors, including aerospace, chemical, biomedical, sustainable energy, and oil and gas. Apart from his industrial experience, he taught engineering students applied dynamics for several years before starting Continuum Blue. His most recent area of multiphysics research in the biomedical sector has focused on fatigue analysis of a spine implant, which has been approved in Europe and granted a U.S. patent based on its novel material technology. And so here's our agenda for today. I'll begin with an introduction to modeling with COMSOL Multiphysics and the simulation capabilities in COMSOL that are sure to benefit biomedical researchers. Then Mark will take over with the main presentation today, featuring his work on simulating spine mechanics, orthopedic implants, and bioreactors. We'll have a live demonstration in COMSOL that highlights fluid dynamic simulations with relevance to a bioreactor example in Mark's presentation. Finally, there's the Q&A session, followed by a brief how-to. So throughout the webinar, we'd like to encourage you to submit questions, and you can do this simply by typing them in the chat window as part of the GoToWebinar controls. You don't have to wait until the Q&A session starts at the end to submit your questions. We'll take a look now at simulation in the COMSOL environment. COMSOL is a multiphysics as well as a single physics simulation platform, and it covers mechanical, fluid, electrical, and chemical simulations, or really any physics that you can think of. COMSOL is particularly well known for multiphysics or coupled phenomena. By this we mean two or more physical effects that interact with each other and without limitation to the number of physics that can be included in a model. You can use the built-in physics interfaces available with COMSOL Multiphysics or define your own custom physics. You can combine, for example, many physics, including electromagnetics, heat transfer, structural mechanics, and fluid flow, enabling the simulation of complex multiphysics phenomena such as Joule heating, thermal expansion, convective cooling, RF heating, and fluid structure interaction. Any or all of these could be present in biomedical applications. On this slide, we demonstrate FSI at left in the figure, and at right, an example of a heart valve simulation that requires it. The heart valve example was presented previously by an attendee at the COMSOL conference. With COMSOL, you'll also find there are many benefits to staying in the same environment, even for single physics simulations. This allows you to use the same workflow no matter what the task is. And so in this sense, COMSOL is truly a unified simulation platform that enables cross-disciplinary product development. Next slide. So let's briefly explore the COMSOL desktop. Our interface is quite straightforward to use, where all the modeling steps are recorded and can be edited later on if needed. This actually happens in the window referred to as the model builder. We input or select our custom settings in the window just next to the model builder and then visualize our results in the graphics window. Clicking on a specific physics node in the model builder allows you to view all relevant parameters and equations in the settings window. It is also possible to evaluate and couple several model components in the same file. As shown here on the home toolbar, we also provide a model library with many helpful example models that are relevant for simulating biomedical designs. Just in case you're curious, the model shown in the graphics window is from a transient study of electrokinetic flow. It is available in the model library. Next slide, please. And here's the COMSOL Multiphysics product suite. All products can be used together to create many unique physics couplings. The products marked in blue represent physics and application-based modules. Those in green are multi-purpose. And in yellow are products for interfacing with different CAD formats, software packages, and programs for data organization and visualization. Next slide. Well, you've now seen our product suite. It's fair to say that pretty much any module can be used for simulating biotechnology because of how diverse it is. Of particular interest, though, is the ability to perform structural analysis, including for nonlinear structures, or even incorporate poroelastic material properties, particularly helpful for simulating tissue systems, such as the artery shown in the figure at top. In COMSOL, you can simulate many types of fluid flow, microfluidics, and acoustics. You can also add mass diffusion and chemical reactions. Thermal analysis can be performed readily, as can electromagnetic and joule heating. There are built-in physics interfaces available in COMSOL for this. I show an example here as it applies to tumor ablation in the bottom figure. COMSOL also has particle tracing capabilities through the particle tracing module and has been featured in a prior webinar for those who may be interested. We recommend that you go to COMSOL.com, and when you're there, look for COMSOL News. It's accessible from the home page. This is a free online resource where users from all industries talk about how they use COMSOL Multiphysics, so make sure to check out our latest issue. I just wanted to share a few simulation examples with you, and so I chose a few from COMSOL users featured in COMSOL News this year. On the next slide, the first example in this study by Boston Scientific, in which they are using multiphysics simulation to better understand the release mechanism in drug-eluting stents. After insertion into an artery, drug-eluting stents release medicine into surrounding tissues to help prevent restenosis due to tissue growth around the inserted stent. The researchers developed a pore shell model of the stent coating in COMSOL. In their work, design parameters that cannot be directly measured, in this case polymer shell thickness, were refined using the optimization module to obtain a best fit between simulation results and experimental data. And so on the next slide, I have just one more example for you. This example features Biotechnology from Biomedical, where they have used multiphysics simulation to optimize their system for hematology analysis. The ABX Pentra series of analyzers uses a combination of optical and electrical impedance measurements to sort and count cells. Hydrodynamic focusing in the device improves measurement accuracy. COMSOL is used primarily to improve the electrical impedance system for cell counting in their Pentra series of hematology analyzers. They set up their model geometry by importing a CAD drawing. Inside the test chamber, a combination of complex physical processes, including high fluid velocity, pressure drop through the aperture, heat transfer, and intense electric fields complicate the design. COMSOL is used to better understand how these physics interact within the device. Now, I'd like to turn the presentation over to Mark, who will share some of his own examples and experiences in using COMSOL for biomedical applications. Mark?
Great. Thank you very much, Jennifer. Hello, everybody. My name is Mark Herman. As Jennifer mentioned earlier, we're a company from the UK, and we're COMSOL certified consultants. Just to give you a brief overview of what we do and the services we provide, we do multi-physics simulation using COMSOL solely. We also do material selection and optimization work for clients, material testing, failure analysis, design optimization, and contract research. As you can see in some of the figures there, we're in a beautiful part of the country, and we have quite a diverse field work from wet and dry labs to simulation work and orthopedic medical implants. Just to give you an idea of the sort of topics I'm going to touch on today, and just remember that all these topics, the simulation work has been done in COMSOL. So on the left-hand side there, we're going to start off by discussing, or briefly going through some bone remodeling work, which involves a total hip replacement, and that involves structural and partial differential equations. Then we're going to move on to bioreactors and work on some cell cultures, a multi-phase flow, and illustrate how easy it is to implement multi-phase flow and two-phase flow models by doing a live demo of a simplified model. And then finally, we're going to touch on some spine mechanics that we've done on a diseased spine, a spine with spina bifida, and that involves structural and fatigue work. Right, to start off, as I said, we're going to start discussing total hip replacements and bone remodeling. And this work involved a company called Corin and another company called iMorphix. Corin is a medical implant company that develops total hip replacements and other orthopedic implants. Just for those that don't know too much about total hip replacements, the main issue is obviously if you get a total hip replacement, be that because you've got diseased bone or cancer or something like that, the implant, once put in, has a problem with aseptic loosening. That means basically the bone around the implant either mechanically fails at the bone-implant interface, or there's a chronic foreign body inflammatory response. And this is normally due to implant wear debris, where you've got this hip replacement, the ball joint rubs against the cup, and you get these little wear debris coming out of it. The other issues with aseptic loosening are relative micromotion between the implant and bone. So, for example, if the implant hasn't been set correctly or implanted correctly, there might be small amounts of motion, which can cause long-term degradation of the bone and obviously patient pain. And then finally, the result of this sort of thing is stress shielding, and this is basically a response or reduction of the bone mass around the implant due to the redistribution of the loads in the implanted bone and a change in stimulus to maintain that bone mass. And that's what we're going to touch on today, this bone remodeling or stress shielding response. Now, don't worry, this is a very busy slide. I'm not going to describe it in too much detail. But essentially what bone remodeling is, it's due to a stimulus, and that's represented by this graph here, which is the red line and the dotted red line. And the horizontal axis represents a strain stimulus or load stimulus. Okay, and the vertical line represents bone resorption or bone loss and bone formation or bone deposition. So if you're in a certain area in that graph, so for example the left-hand side as shown by my mouse, this is where bone loss occurs, and that's because the loading on the bone is lower than what it normally would be. In between this lazy zone, you don't get any bone remodeling. Okay? In other words, your bone stays static, there's no bone growth or bone loss. While in this region here, we get bone growth. This is where you've got excessive load where there didn't used to be load, and so what the body does is lays down bone material to compensate for the loading of the bone. Obviously, if that strain stimulus goes too far, what you can do is obviously get cracking or damage, which is excessive damage, due to bones breaking and the like. So when we do these sort of bone remodeling studies in COMSOL, what we do is actually set up two types of models simultaneously in a single COMSOL model, and that's an intact femur. That's one where you don't get any implant, but yet you do the same sort of bone cut lines as a reference point. And then you've got an implant model which is shown on the right-hand side, and that's obviously where we put the particular implant in. In this case, it's showing a short stem total hip replacement for the femur. And that's just so that these models are of the leg bone. Okay. With the COMSOL, the great thing is that it's pretty easy to implement loads and kinematic information. And you can use dynamic loads or static loads. In this case, we applied kinematic loads from a reference Bergman et al. From 2001. And this is based on patient data that was, they measured 4 patients, quite a range, and they measured the weight and the loads on their body while they were walking or stair climbing. Typically, elderly people are the ones that suffer and require total hip replacements, so this is where we analyzed for particular use, where they're walking, or the other harsh loading condition for elderly people is stair climbing. However, what we do is in the model we can apply these loads in a particular way, and obviously the right-hand image there is the pelvic bone, which shows the sort of gluteus maximus or muscles around your pelvic region, and their attachments to the femur or leg bone. And obviously we coupled those up and showed the blue regions are where we apply the loads over a cycle. One of the most difficult things is trying to get material properties, especially when we're looking at patient-specific data. So in this case, we're looking at a very specific patient that required a total hip replacement. And obviously what we do first is we take a CT scan of the patient's region, of the disease region where the implant is required. And then we've got to convert those to material properties that COMSOL understands. And in this case, what we did is CT scans come in representative grayscale representation as shown in the left-hand image, and that's called Hounsfield units, or HU. And depending on the scale of the gray region or the color, you can get a function of density, or density is a function of Hounsfield units. Additionally, obviously, you can make Young's modulus a function of density. The two illustrations or plots on the right-hand side show the density mapping of this patient-specific bone. And you can see there's quite a density variation along the leg bone from a very low value of about 10 kgs per meter cubed up to 2,200 kgs per meter cubed. And it shows the regions with high density and low density. Obviously, one of the things we have to do with these material properties, we have to validate the materials. And this includes, we took a validated femur model in publication, and we mapped our patient-specific data onto their density and Young's modulus distribution to ensure that we had a good representation of the material properties inside the femur or leg and pelvic region. Just bear in mind, we're never ever going to get the exact same density distribution or modulus or Young's modulus distribution, as these are, our model is a patient-specific model and the validated femur was a different patient. Okay. To give you an idea of the implants utilized in this study, we used two types of implants from Corin. One is a mini hip, which is what they call a short stem, as illustrated in the top right-hand image, and the other is a metafix, which is a long stem, so this goes quite far down into your bone, and that's shown in the right-hand lower image. Now, the difference is, the preference is to go for mini hips or short stem implants, because these are bone conserving types. In other words, you don't have to chop up, or the surgeon doesn't have to chop out so much bone to put these in compared to the metafix or long stem implants. However, you get a very different response from these two types of implants, especially when it comes to bone remodeling. And ideally, you want these implants to last for 20 to 30 years in the patient and not have to take them out after 5 or 10 years because of aseptic loosening. In terms of the relative size and position and resection of these two types of implants, there's different sizes of mini hips. So you get mini hip 1, mini hip 2, 3, 4, and 5. As you go from the lower numbers to the larger numbers, the mini hip design gets slightly larger, and that's for different types of patients. So a large person will get a larger mini hip 5, and the cut lines, as illustrated by the dotted lines here, will be slightly different depending on the type of implant you put in. So this is just showing how the surgeon would implant them in reality if the patient was having that particular type of implant. In this particular patient, the ideal mini hip size was a mini hip 2, while the metafix size is ideal in this particular case. What we did is we ended up loading the implant and having a look at the load conditions. Now, the great thing with COMSOL is you can define where you want strains or extracting data in graphs within COMSOL. In this case, we selected a blue line along the medial line, or the inner leg of the hip, or the inner section of the leg, and we took a line and we plotted the strains on that bone as it was loaded under walking conditions. And here you can see the variation in the types of implants and this, I've got MH represents the mini hip, MF represents the metafix, and obviously the numbers represent the size. And the light blue line represents the intact model. the comparison of the implants is relative to the intact model. Now, in the ideal cases, you want the implant to respond exactly like the intact model. This will prevent the bone remodeling response observed in the bone, and therefore loosening or deposition of bone in different regions of the bone, which you don't want. So the further away these curves are, for example, the red line, which is the Metafix 2, away from the blue line, the worse the bone remodeling response. Okay, so obviously in this particular case, mini-hip 2, the brown line, is actually a better model in terms of response inside the body. However, we did this study and we found something very interesting. Is if the surgeon doesn't put the implant in the right position or malpositions the implant, you get a very different response. So this is the black line overlaid on the graph seen earlier is basically mini-hip 2 malpositioned by the surgeon. And as you can see, it's diverged away from the original brown line, where that's the ideal location in the surgery, and it's even further away than the blue line. This illustrates that in the study that we found that actually surgeon response is quite important, or surgeon positioning of the implant is very important to ensure that there is a good take-up of the implant and no aseptic loosening, long-term aseptic loosening or bone remodeling. So this is the great thing about COMSOL, is that you can do all this inside COMSOL, and you can do it real time. So for example, if you're interested in doing patient work and you need an operation tomorrow, you can actually do this study overnight and assess which position to lay the implant is best. And here's just some illustrations at different time points of the leg bone and how it responds over time. So for example, the red represents bone deposition, the blue represents bone loss, and you can see on the left-hand side the Metafix. The Metafix is the long stem model. You get a lot more blue regions, which means a lot more bone loss, and less bone red regions or bone deposition. Unlike the mini-hip size 2 on the right-hand side, there's a marked reduction in bone loss. You are going to get some bone loss, but this shows how much better this mini-hip or the smaller size implant is. The great thing is you can do all these plots inside COMSOL, these, in terms of finding out where you get deposition over time, and you get time-dependent responses. I've also just shown this for additional response, that obviously with the change in the implant, you also get a change in the pelvic region, where you get bone loss and bone gain in the pelvic bone, in addition to the leg bone due to an implant. And this is just different time shots at 1, 3, 6, 12, and 24 months. Okay, so that's the sort of capability inside COMSOL for bone remodeling. I'm now going to go on to another topic, bioreactor optimization. This is some work we did with a company called Selexis, who's developing a new bioreactor system. Just to go over the bioreactor system they're developing, it's called the Cell Maker, and they've got two versions, a regular and a plus version. The great thing about these bioreactor systems, they're single-use disposable bioreactors, not the equipment that you see on the illustration there. That's the control system for the bioreactor, but the bioreactor bag, which is placed in that system, is disposable. It's a state-of-the-art system which is easy to use and completely fully automated, and it includes a sort of wealth of technologies for growth of a wide range of cell types, including E. coli, algae, bacteria, and the more challenging mammalian and insect cell lines. Okay. The system essentially uses a simple scalable airlift technology and doesn't use any mechanical mixers. Just to give you an idea of how this is a schematic of the bioreactor and the parameters involved, a bit of an overview. So essentially, this is a side view of the disposable bag, and you've got a little tube, a sparge tube at the bottom where you pump gas in, carbon dioxide, oxygen, and the like, and you bubble air through the system, so that's through a vertical column, and you see the bubbly flow with the red arrows, and the blue line represents how the medium, or your mammalian or cell type, moves around inside your bioreactor. As the bubbles reach the surface at atmospheric pressure, the gas is drawn out and or recleaned and charged again. So that is essentially the bioreactor model that they had. Now the issue they had with this is they had certain cell types dying off rapidly, especially the mammalian, difficult mammalian cell types. And so they wanted to optimize the bioreactor design to optimize the bubble size and the bubble flow conditions to maximize cell or minimize cell death and maximize cell growth. So, okay, in terms of the work we did for Selexis, what we first did is we had to sort of assess what types of what type of bubble sizes and the flow rates that were coming through their current design system. So what we ended up doing is doing some high-speed video footage of the bubbles coming through the bioreactor over time. And this just illustrates some of the bubbles coming through the system. So you've got a single port and a multi-port sparge tube at the bottom of the bioreactor. And what we ended up doing is taking high-speed video camera footage of the system to assess the size and scale of the bubbles as they rose through the bioreactor column. And this was mainly to validate our CFD model developed inside COMSOL. Hopefully you can see some of the animations coming through this moment. As you can see the bubbly flow coming through. And we monitored these bubbles over time. And what we ended up doing is graphing the physical data, the equivalent circular diameter and the aspect ratios. The equivalent circular diameter, so what we did is as the bubble was moving up the column, we tracked it and analyzed the area or cross-sectional area of the bubble as it moved up. And as you could see from the animations earlier, they sort of wobble as they're going up and they change shape. So as it was coming out, we assessed the aerial in 2 dimensions, and then we found an equivalent circular diameter. In addition to that, we found the aspect ratio, which is the longest length over the shortest length, and assessed that over time. And here it just represents a graph of 3 different types of data sets from different studies we did in terms of flow rates, height of the column, positions of the holes, and locations of the holes. And you can see there's quite a diverse data set there in terms of equivalent circular diameter. Now the idea was to then model these and ensure that our CFD model was validated. So what we ended up doing is doing a number of runs with those particular data set and ensure that our models were in line with what was coming out of the physical tests. As you all know, or I hope you all know, that finite element analysis or finite element models need to be validated against something. And this is the validation stage. Obviously, it's not always possible to do validation of the models, and sometimes you have to sort of use finite element analysis as a comparative study to find out which type of design is getting better or which is getting worse in certain types of parameters. But it won't give you absolute values. In this case, we validated the models. We did some modeling, and we did some correlations to the high-speed physical tests. And here's 2 sets of data that we did some comparisons to. Data set 1 and the model 1, and data set 2 and model 2. And as you can see, in data set 1 we get a bit of a divergent response. In data set 2 we get a better or very close correlation. However, we were very happy with the response over several sets of data, that we were quite confident that the model was predicting the physical response quite accurately. Now the question is, if you're going to optimize a bioreactor, where do you optimize the bubble sizes to? What sort of equivalent circular diameter and aspect ratio are you looking for? So we looked at some publications by Christie and Godet et al., who looked at sparge bioreactors, and they found that most of the damage done to cells was done on the surface. So what they found in their studies is that they need bubbles of diameter greater than 7 millimeters but less than 20 millimeters to prevent cell death, with an ideal target of around 10 millimeters. So the red region in this graph here, or the shaded red region, shows the target area of where we wanted the bioreactors bubble size in terms of equivalent circular diameter and aspect ratio to be within. And that's what we ended up doing. We ended up building the finite element model and parameterized a number of items. In this case, we looked at the number of flow ports, flow rates, the gas mixture. You could change the ratios of oxygen to carbon dioxide to nitrogen, which changed the flow characteristics. We also looked at the head or the fluid height in the chamber or the cell culture height, and some other geometric parameters as illustrated in the schematic in the right bottom corner. So these are the sort of parameters we optimized for, and the idea was to make sure that we moved those bubble sizes and equivalent circular diameters down to below 20 millimeters but above 7 millimeters. And just to give you some idea of the trends that we observed, we found that obviously if we reduced the sparge tube diameter, we found that actually the equivalent circular diameter was reduced. So we knew how to move the equivalent circular diameter to a lower value or increase it in the opposite direction. Also knew how to increase, if we changed the port angle, we could move the equivalent circular diameters, reduce the equivalent circular diameters, also change the aspect ratios. And obviously if we changed the number of ports, we didn't find much of an effect there, which was quite surprising. Anyway, after a number of simulation runs, we ended up finding an optimized parameter set. So of all those parameters that we varied, I think we ran in total about 62 models. We ended up getting a model that was giving us very close correlations to ideal optimized solutions. And we're currently building the bioreactor with these particular parameters to actually physically test and assess with high-speed camera footage. Okay. Now we're going to the live demo, and hopefully with the problems with sound and the lag and response time, that this will actually work. Okay. What I'm going to do today is demonstrate the a rising bubble problem. In this case, we're going to be looking at a bubble of oil inside a column of water, and obviously you've got a film of oil at the top where the bubble of oil goes into. And in this case, I'm going to, and this is sort of applicable to bioreactors, except you wouldn't have oil bubbles rising in the bioreactor, you would have air or gaseous bubbles rising to the top. But this is an example that is in the COMSOL suite, and you can actually just look for it. If you go to File, Model Libraries, and you do a search under there, and you look under CFD or bubble flow, you'll be able to find it. As Jennifer said earlier, the great thing with COMSOL is it's quite a simple, although very complex in terms of what you can do with it. It's a nice layout, neat and nice layout. On the left-hand side here, you've got your modeling tree where you can define all your physics and parameters. In the middle, it's your section or selection response. So, for example, if you want to define parameters, you click on parameters, and you have a section in the middle here which then defines what parameters, and you can type in any type of parameter you want in here. For example, fluid flow. And you can define the units in square brackets. So you can define whatever units you want. So in this case, I'm going to say 10 meters per second. You can easily change units here, so that's great. Then On the right-hand side, you've got a graphic window. The upper right-hand side, you've got your graphic windows, which represents your geometry or the physics domains you're modeling in. And the lower right-hand section, you have your progress bar, messages, and log. When you're running simulations, that becomes important. Obviously, if you don't like the layout, you can actually easily quite move these windows around to your preference and use, especially this becomes useful if you've got multiple screens and you like working across a number of screens. Now, in this particular model, I'm going to make use of what they call 2D axisymmetric modeling. And if you want to build any kind of physics inside COMSOL, what you do is right-click at the top here, and you've got a selection of different domains. So you can look at 3D modeling, 2D axisymmetric, 2D, 1D, or 0D problems. In this case, we're going to be looking at a 2D axisymmetric. So typically, you just click on 2D axisymmetric, and you're able to define your model. And this is what would come up on your model. And in this case, we're going to be looking at this particular geometry here, which represents a 2D axisymmetric region of your bubble, which is down here, an oil bubble, moving up through a column of water, which is this section here, up into another region of oil at the top here. Okay. Now what we've used here is used laminar two-phase flow level set method, which is a method of defining two phases. You've also got phase field as another option inside COMSOL. You can obviously also change this to turbulent modeling and the likes. So if you click on this particular tree, you can see that you can change the method here if you want to look at different types of phase fields or multi-phase flow for those who are advanced users. You can look at different types of incompressible to compressible flow if you're looking at gaseous bubbles and the like. So in this particular case, if you want to look at different types or add different types of models, you just simply right-click on the component. You can add physics, which is very simple, and add additional physics nodes if you just don't want to look at fluid flow, but you also want to couple it with thermal responses or the like. So I'm not going to do that today. I'm just going to sort of illustrate some of the features in this particular model, and I suggest that you go onto COMSOL's website. There's a number of model examples, or ask COMSOL for a trial license to actually look or use COMSOL and get used to it and have a look at some of the tutorials. When you click on the physics node, you'll see that it gives you a list of little nodes that represent the physics. I'm just going to quickly group this by space dimension. That's how I like to present my models. And here you can now look at what are the domains. You can look at fluid properties. And in this case, we're looking at two types of fluids. And in this case, we've defined a transformer oil and water, the two fluids. And if you go up to the tree, you've got a section here which defines where your transformer oils are under the materials section. You can see I've defined domains 2 and 3 as transformer oil, and domain 1, which is, as I discussed earlier, as water. Now, in this particular case, you can look at different sections of CFD flow and look at different types of flow characteristics. So here you can ignore surface tension between the two phases by just simply going down this column and clicking on the neglect surface tension. Obviously, we're going to keep it active. You also have a library of different types of gas and liquid interfaces or liquid-liquid interfaces. In this case, it's a liquid-to-liquid, and we're going to select the olive oil to water surface tension characteristics. You can also do your own user-defined characteristics, or you can, if you want to look at air bubbles with water, you can change those as you wish. So it's a really nice, neat interface to change characteristics within the model. And then it's simply defining your initial values of each domain. So in this case here, I've defined domains 2 and 3 as fluid type 1, which we've defined in the fluid properties here. And if I click back on fluid 1, it's transformer oil. So, and likewise, I've also defined the initial values of the water region as fluid type 2. So this defines your two regions of fluid types. In addition, obviously, gravity plays a role here. So we've added in a gravity constant in the Z direction, and that's just to look at the gravitational forces. On top of that, obviously, we've got to look at boundary conditions. So we've applied the axisymmetric boundary condition on this edge here. We've also looked at walls at the top and the bottom. However, you'll see that on this wall section here, I've defined it as a no-slip boundary. So there's no forces, drag forces on the surfaces. You also define where your initial interfaces are between your phases or your gas and air or water. In this case, it's oil and water. Additionally, on the outer column, where we want the bubbles going up, we also have a wetted wall phase, and you can change your contact angle or slip length. Okay. So it's very easy to define characteristics in here. If you want to add additional nodes, you simply right-click over each section, and it's very, very simple to add in different types of physics and characteristics. Obviously, with all finite element analysis work, you need to mesh the model. In this case, it's just a simple mesh, and we just did a time-dependent study to assess as this oil droplet rose to the surface and came through the surface, similar to how the bioreactor worked with the air bubble coming to the surface and looking at how it responds. Now, I've already run this study because it's quite hard computationally, but I ran it on my laptop, and it took about 18 minutes to run over a full period before the bubble reached the surface and broke the surface. So I'm not going to show you the study. I'm not going to run it now. I'm just going to show you that it's pretty easy to set up steps inside your study and look at a time-dependent study. And this study was run over 0.5 of a second. Now, the great thing about COMSOL is it's pretty easy to add in various plots. So here's a solution at time 0, 0.08 seconds. And you can move forward at different time points and plot those to look at the bubble as it hits the surface. In this case, I've moved it too far forward. It's already broken the surface. This one's getting close to the surface. And there it breaks the surface. Okay, so you can easily sort of look in your results plots at different time points. You can look at different properties and parameters. With 2D axisymmetric models, you can also change it to a 2D axisymmetric view by simply looking at your data set and adding a 3D view within COMSOL. It's very quick and easy. And obviously, once you've got it running and you're happy with the results, it's very easy to just quickly play an animation inside COMSOL. So I'm just going to show you this quick animation. Hopefully it will work. There may be a bit of a delay. Okay. Unfortunately, the animation's not working too well, so I'm gonna switch back to the presentation as I'm going to be running out of time. So that's the live demo, but however, I suggest you contact COMSOL and ask them for a COMSOL license, file license to test COMSOL out and play with some of the modules and tutorials. I'm now going to go back to the presentation and continue on with the last section of my presentation, which is looking at spinal mechanics, and specifically spondylosis and spina bifida, which is a disease of the spine due to injury or fracture of the vertebral body. The vertebral body is the bony part inside your spine. So just to give you a quick overview of what spondylosis and spina bifida are, basically spondylosis is defined as a defect in the pars interarticularis of the vertebral arch. What is that? Basically, these bony little bodies that protrude out the back of your back are these little pars interarticularis, and the parts that attach those bony bits to the main spinal column. And this is illustrated in this image here of your spinal column, and the red region highlighted is the lumbar region. And that's an X-ray of somebody who's got a fracture, a stress fracture, in these pars interarticularis, and as illustrated in the image. Now these fractures or these defects are typically caused by stress fracture of the bone, and this is especially common in people that are overtrained in sports activities. Yes, you know who you are. If you go too far on your training schedule, you can have one of these, but I must mention that they're mainly in adolescence. Okay. Now, there's spondylosis is commonly associated with something called spina bifida occulta. This is where there's a slight deformation or deformed bone structure in some people where the bone, these little bony bits that protrude at the back, don't fuse or form together during birth or development inside the womb, or there's a space between them. So what we want to do is find out if there was a quick association, if we could model this association inside finite element analysis. So what we did is we built up two models, a natural and an SBO model of the spine. As you can see on the left-hand side, it's the natural model, and those protruding bits of bone in the top image are all there, and there's no deformation or disease section. The SBO model is represented on the right-hand side, and I've highlighted the red region where you're missing bone. So that's where there's some spaces there that's due to the malformation of the bone during in the womb and development of the baby. So what we simply did in COMSOL is we imported spine models from a patient-specific scan, similar to how we did the bone remodeling earlier on. And we simply applied a vertical load and a rotational rotation. So this is somebody that's running along and jumping up and down and then turning at the same time, such as seen as in a fast bowling cricket. So we only modeled the L4 to S1 components of the lumbar, and we actually loaded the lumbar section up to 1 kilonewtons and a 3-degree rotation. And then what we did is we assessed the stresses on these two sides, the left and right-hand side, the ipsilateral and the contralateral, sorry, medical terms, or if you want left side and right side, to assess what was happening in the loading conditions, and had to look at the L5, which is your lumbar vertebra 5, to assess fatigue, failure, and stress occurring in that region. Again, we did some validation work of our model and the patient-specific work, and we looked at other people in literature. Here we showed the comparisons of our model versus some 2 sets of data available in literature for range of motion. And we also did facet contact forces. The facet points are where the two vertebral bodies make contact, and you get some slipping and sliding as they rub against each other. So we had to look at the forces across those. The great thing with COMSOL is you can easily model contact sets, which is a very difficult structural mechanics problem in itself. So once we had sort of built the model, we simply looked at the differences between displacements and stresses and fatigue responses of these types of models. And you can see straight away there is quite a dramatic change, as you'd expect, especially if you're missing a section of your bone. So on the left-hand side, you've got your SBO model, which shows a lot more displacement in certain regions, while you've got your natural model on the right, which shows a little bit more or less motion and more rigidity. But when we looked at these two sections, the left and right regions, we found a dramatic change in stresses. And this is the stress amplitude during motion, left and right motion. And here you can see that the blue line represents the intact or ideal case, and the red line represents the red case. And you can see there's a dramatic section of increase in stresses, double the normal stresses. We then modeled this and compared it to bone fatigue properties from some papers and literature to find out after how many cycles or how many times a cricketer, for example, could bowl before he got a spondylosis or fracture in his spine. And we used the work of Zupos et al. In 2007 to get these curves of different types of bone loading conditions. And what we then did is use COMSOL to estimate the fatigue failures under repetitive axial loads and rotation conditions. And what we found is that you've got a dramatic number of cycles before you got stress fractures inside these regions in the bone, up to 70,000 cycles, while the intact case lasted up to 10 million cycles. And this correlated very well to clinical studies, where we got very much similar results for people with SBO. We found that they did have very early indications of failure. Okay. And the great thing is it's very easy to use COMSOL to implement these studies and assess where failure is going to occur with their fatigue module. Right, well that ends my presentation section. I'd just like to acknowledge some people in the work for the total hip replacement bone remodeling, coronary morphics, the Technology Strategy Board, and the work done on the spinal bifida and spondylolysis, Nottingham University and Nottingham University Hospital. Thank you very much, everybody, and now I'll pass you back to Jennifer, which I hope is there.
Yeah, so thank you so much, Mark, for your presentation and demonstration. So, yep, it's time for the question and answer session. So the first question that we have today for you, Mark, is how long does it take to perform a complete bone remodeling analysis? And typically, what kind of hardware are you using to run this analysis?
Okay, typically they are very long simulations, because you can imagine with the bone mappings that they take roughly about 22 hours. But the hardware we're using isn't that excessive. It has got a lot of RAM, 192 gigs of RAM, and it's using 2 Xeon processors. But we always find that it sort of uses only about 32 gigs of RAM. And it handles the sort of large models quite happily with over 1 million degrees of freedom.
Okay, our next question. How did you implement the elemental bone material properties from the patient CT scan in COMSOL?
Okay, as I said earlier, the sort of CT scans come in what they call Hansberg units, which are just literally coordinates with values or HU values. So they have an X, Y, Z value and a point with a grayscale value. And we literally imported those text files inside COMSOL. And it's very easy to do with a function. So you simply implement a function inside COMSOL, and you say that the first 3 columns of that function represent your X, Y, and Z coordinates, and your 4th column is your grayscale value. And then we called in that function and then converted those grayscale values to density and Young's modulus values with a functional relationship defined by literature.
Okay, thank you, Mark. Our next question, is it possible to simulate osteoporotic bone in COMSOL?
Yes, it is. It is possible. It's the thing is you have to sort of obviously get a very refined model and region. So, for example, our material mappings are based on CT scans. Some of them are great, some of them weren't. Sometimes you need micro CT scans to get that sort of high resolution detail. So basically, you've got to get your X, Y, Z coordinates and your point valuations very close together to assess your low density points and your high density points. But yes, it is possible.
Okay, great. Our next question, is it possible to model the large-scale bioreactor systems such as effluent plants and industrial wastewater treatment in COMSOL?
Yes, but I wouldn't suggest using the model that I just demonstrated where you actually model the bubbles themselves. COMSOL has some bubble flow models which simplify the modeling process, and they do a volume fraction of air to water ratios, because as you can imagine in those sort of affluent plants and things like that, there's a massive amount of bubbling and processing that it would just be computationally too excessive to actually try and model it as individual bubbles. So I would suggest using the COMSOL bubbly flow models that they have, and you can get them in terms of laminar and turbulent physics interfaces. So yes, you can.
Okay, great. The next question, was contact modeled in the spine and hip replacement models? If so, was friction also included in the models?
Yes, yes, they were. They were with friction across the ball joints between the cup and the stem head, so the ball section. But we didn't model friction in the action between the implant and the bone along the whole region, only sections of the bone where there was titanium beads or a rough surface.
And so we'll do another question. In regards to the spine model being imported, what format of models can be imported into COMSOL?
Okay, you can use, obviously if you are a regular user of 3D CAD models and suites, you can import from different modules like SolidWorks, AutoCAD, Pro Engineer, CATIA. So those are sort of your typical 3D CAD import types. With our particular case, what you can do is you can actually use two types of methods. Either you convert your CT scans or your patient-specific scans into SDL surfaces, so you define the outer surfaces of your bone. So between your soft tissue or muscle and the bone, you define those surfaces by SDLs, import those SDL files as 3D geometries, and then implement the mapping, which is the grayscale Hounsfield units that I mentioned earlier from your CT scans, and you overlay your density distribution and your material properties, overlay that into your domain or your 3D domain. The alternative is just import your mappings, and you define where you've got very low or soft tissue regions, you define those as voids or low Young's modulus, extremely low Young's modulus values. So they're not actually structurally important. So you've got two methods if you're looking at patient data, or you can develop the models in 3D CAD packages and import those directly inside COMSOL.
Okay, fantastic. Thank you. And so just one last quick question. Can COMSOL be used with HPC?
Yes, it can. Yeah, there's a number of things that COMSOL doing, and you can actually use it with Amazon Cloud services or EC2 services, or if you're building your own in-house cluster, you can use them on there. Just bear in mind you need a particular type of license. You can't just have a standalone CPU license. You need a floating network license to run HPC types of studies.
All right. Mark, thanks so much for answering all of these questions and, of course, for your presentation. So I'm going to wrap up the webinar. So on the next slide, a question that we hear quite often from the audience was about how to try out COMSOL Multiphysics. And one way is to join us at a workshop. Our workshops are free hands-on events where you can meet a COMSOL specialist and try COMSOL Multiphysics with their guidance. Next slide. We'd also like you to know that online registration is open for the COMSOL Conference 2014. This will take place this fall at locations across the globe, including in the U.S., Europe, South America, and Asia. So make sure to save the date and register if you haven't already. Our conference includes over 30 hands-on mini-courses, demo stations, and presentations by COMSOL users, making it an excellent way to get in-depth information about COMSOL Multiphysics and its many capabilities. This year, there will be a mini-course introducing the Application Builder, a new tool that will be available in COMSOL 5.0, which is going to be released very soon. For more information about the conference, to submit an abstract, and to register, please visit www.comsol.com/conference. And on the next slide, you'll also find helpful information about multiphysics and our products on our website. And there's more. You can find user stories, a model gallery, the COMSOL blog, and a discussion forum. You can also contact us at comsol.com/contact. That's it from us today. Thank you, everyone, for your attention, and enjoy the rest of your day.